In this experiment, I am not just counting the frequency of the individual codons. I'm going to count the frequency of strings which consist of 3 codons and see if it matches the frequency of the reverse complement of that string. To do this, I have created some software that searches for 64 separate strings and their complements. For example, TTT, TTT, CCA, TTT, TTT, GTA, and so on. And it will search not just for this string, but for its complement within the human genome and see if the frequency of any one of these strings is equal to the frequency of the complement. So we moved on from just counting the frequency of an individual codon such as CAT, and we're now counting the frequency of a 3 codon string and seeing if it matches the frequency of the reverse complement of that entire string. So click on complement. Now as you can see, we're scanning just chromosome number 10 of human DNA. This is the number of lines scanned, each line consisting of 70 letters or 70 bases. And here you can see the count for the string and the count for its complement. Now, so far you can see a rough correlation. So it goes 22, and then this goes down to 2, and that goes down to 7. Then it shoots up to 21, and that shoots up. And then that shoots down to 1, and that's 1. Then this shoots up to 20, and that goes up. Then that goes down to 4, and that goes down. This goes up to 189, 200, and that goes up to 230. That goes down to 44. So you can see that there does appear to be a consistent correlation between the number of times any particular string of codons occurs and the number of times that the reverse complement of that string occurs. The consistency of this finding over 64 random codon strings suggests that the occurrence of a particular string of codons must be determined by the frequency of the individual codons that make it up. And because the individual codons are all complementary on this strand to this strand, and as we've seen, complementary codons have the same frequency, so the total frequency of the entire complementary string of 3 codons is always going to be equal to the total frequency of the 3 codon strings in this column. So in some way, the frequency of each individual codon is determining the frequency of the whole string. Even though the strings, the complementary string is reflecting the number in the first column. As you can see, the ratios are in many cases not equal to 1. 0.8, 1.6, 1.2, 0.5, 3.6, 2.8, and so on. So there isn't an exact equality, but there is a kind of reflective approximation. What this experiment therefore has demonstrated is that the frequency of any string of codons is equal to the frequency of the complementary string. it's finished now scanning chromosome 10, so we're going to copy the frequencies of the codon strings and their complements, and we're going to paste it into Excel. So we need to create a new spreadsheet, and what we're going to do then is simply look at the line chart for that. Now if we make this bigger, you can see how close the correspondence is between the frequency of a particular the codon strings and the frequency of their complements. At every point, one is shadowing the other, which is very it's very unexpected. The obvious explanation, or the explanation that first comes to mind, is that because the complementary string is made up of complementary codons, and because we know that complementary codons have the same frequency as the codons that they're complement for, then the codon the frequency of any codon string must be simply the product of the frequencies of the codons that make it up. Hence the matching of the frequencies. The bizarre result of this, though, is that it means that no matter how long the codon string, the frequency of that string will be equal to the frequency of its complement. It's possible, though, that as the number of codons within a string increases, the probability, the coincidence of probability may diminish due to the accumulation of errors. Anyway, that's the end of this video. So